Hello class. Okay, now let us start the new chapter today, that is chapter 6, your seed. We will be discussing in detail about the structure of the seed and its germinating process. Okay, now before going on with the chapter, that is the structure of the seed. Okay, first let us understand three terminology. Let's get familiar with three terminology, that is seed, fruit and grain. What is seed? So our previous chapter was fertilization in plants, right? So we have discussed that after the fertilization, what happens to the ovule? Your ovule gets converted into seed. So your seed is the ripened ovule. Okay, it has a baby plant inside it, or you can term it as embryo inside it, which under favorable condition germinates and give rise to new plant. Okay, now next is fruit. What is fruit? After the fertilization process, your ovary converts itself into a fruit. Okay, your ovarian wall is converted into a fruit wall. So your fruit is the ripened ovary. Now we all know that inside the fruit you have a seed. So what is the primary function of the fruit? The primary function of the fruit is to protect the seed inside. Okay, and it also helps in dispersal. Another term, grain. Now what is grain? Now grain is also a fruit, but here your fruit wall and your seed coat are fused together and they form a single layer. Okay, example of grain are your maize, wheat, all these are the examples of grain. It's also a fruit, but here your fruit wall, we cannot distinguish between fruit wall and seed wall, they are fused together. Okay, so that's all about these three terminology. I hope you could differentiate between all three. Now we will be discussing more about seed. Okay, carrying on further with the detail about the seed, we all know the seed is the ripened ovule, right? After the fertilization, your ovule gets converted into seed. We all know that it has a baby plant inside it, which under favorable condition give rise to new plant. That means germination takes place only under a favorable condition. Okay, now for providing a food or nourishment to that baby plant, you have an endosperm portion, and main function of the endosperm is to provide a nourishment. We know that during fertilization process, one of the sperm nuclei would go and fuse with the polar nuclei which was present in the central cell, thus giving rise to endosperm. And this endosperm, I told you that it provides nourishment to the growing embryo. Okay, now that's all about the characteristics of the seed. Next we will be talking about the types of seed. Okay, so we have seen seeds of a different size, right? Have you seen the seed of apple? Have you seen the seed of a coconut? Do they have a same size? No. So seeds, we can find a seed of a varying size. Okay, now next moving on to the different types of seed. First we will be discussing about the type of seed on the basis of endosperm. Okay, we all know how when the endosperm is formed, right? So on the basis of endosperm also we can divide the seed into two types. Okay, albuminous seed and your ex-albuminous seed. Now albuminous seed is also referred to as endospermic seed, and ex-albuminous seed as a non-endospermic seed. Now first let us discuss about albuminous seed. Now albuminous seeds are mainly present in a monocot seed. Okay, now in this case the major portion of the seed is covered by your endosperm region and your cotyledons are very much minimal. Okay, thin membrane of cotyledon is only present. Most of the region of the seed is covered by a endosperm. This is usually found in a monocot seed. Okay, now example of monocot albuminous seed are your palm, millets and dicot. Exceptions are obviously there, so your dicot seed also can be albuminous. So that in that case examples are your custard apple is one of the example of dicot albuminous seed. So another type is your X albuminous seed, also known as non-endospermic seed. Now in this seed, what happens is major portion is covered by your cotyledon. Your cotyledon becomes thick and fleshy, and they store food. They provide nourishment to the growing embryo. Okay, so those kind of seed are referred to as an X albuminous seed. Okay, now these are generally present in your dicot seed. So example, though there is exception, monocot seed also, some of them are X albuminous, but majorly it is found in a dicot seed. So example of a dicot X albuminous seed are your gram, mango. Okay, seed of a gram and mango are your example of dicot X albuminous seed. And example of a monocot albuminous seed, X albuminous seed, are your orchid and blessinaria. Okay, these are the seeds present in orchid and blessinaria, are also non-endospermic. So it was on the basis of your endosperm being present. If it is present in a, if the seed is covered majorly by the endosperm, then it is known as your endospermic or albuminous seed, and if the cotyledon covers the major portion, it is known as X albuminous or non-endospermic seed. Okay, so those were the types on the basis of endosperm. Okay, next we will be discussing about the structure of one monocot seed and one dicot seed. For studying about the monocot seed, we will be taking the example of maize, and for studying the dicot seed, we will be taking the example of bean. I hope you know what is monocot and dicot seed, right? Monocot means only one cotyledon is present, but in case of dicot seed, you have two cotyledons. Okay, so maize is the example of a monocot seed, and your beans like gram or pea are the example of your dicot seed. So we will be discussing about the structure of both monocot and dicot in detail. Okay, now let us study the structure of dicot and monocot seed in detail. For dicot seed, we are taking bean seed as a study model. Okay, first let us study the external structure of a dicot seed, that is bean seed. Like all the seeds, your dicot seed are also protected by a seed coat. Now in case of bean seed, you have two layer seed coat. The outermost one is referred to as testa, okay, and the inner layer is known as tegmen. Now if you look at this picture given alongside, and if you observe the seed properly that's given there, can you see the whitest scar here in the seed? Can you see that? Now that whitest scar is referred to as hilum. Now what is hilum? Now hilum is the region where your ovule was attached to the ovary through a placenta. Okay, that's the reason where the attachment took place, because we know that ovule only later on gives rise to seed, right? So attachment region of ovule with the ovary is known as hilum. Now just beside the hilum, you have a small pore like structure. That pore like structure is known as micropyle. While discussing about fertilization, we have discussed the role of micropyle, right? What is micropyle? What is the role? It's a point where a pollen tube is going to insert inside the ovule. Okay, it's a region from where your pollen tube is going to connect with the ovule. So this micropyle, other than connecting with the, after the fertilization takes place and after the seed is formed, this micropyle acts as a region through which water seeps inside the seed. Okay, so those were the external structure. Now if you soak a seed overnight, and if next day you are able to open the seed coat, and inside the seed you will be able to visualize a baby plant or embryo. Okay, now your embryo has two regions, radicle and lumen. Radicle will later on give rise to root system. Okay, R for radicle, R for root, so it's going to give rise to root system. And another one, the plumule portion, is going to give rise to shoot system. Okay, So that's all about the structure of a bean seed, that is the structure of the dicot seed. In case of dicot seed, you have two fleshy portion seed leaf, that is also referred to as cotyledon. It's the cotyledon is only going to provide a nourishment to the growing embryo in case of dicot seed. Okay. Now next we will be discussing about the structure of a monocot seed. First we will be discussing about the maize. Okay. Like I told you in your previous class, your monocot seed are generally endospermic seed or albuminous seed because its major portion is covered by the endosperm, and cotyledon is only a thin layer that is present in it. So most of the seed portion in a seed is covered by your endosperm. Okay, now look at the maize seed given alongside. If you can see this oval scar here, it's the region where your embryo is going to be seated. Okay, and other region is covered by your endosperm. Okay, those were the external structure. Now let us get into detail about the internal structure. Now if you do a longitudinal section of a maize seed, maize seed is also referred to as grain. Why is it called grain? At the start of the chapter only I told you that grain means your fruit coat, fruit wall and your seed coat are fused together. Okay, it's forming a single layer, that's why you are referring to it as a grain. Now I told you in a monocot seed, the most of the region is being covered by your endosperm region. Okay, now your endosperm, the outermost layer is covered by a aleurone layer. It's a proteinaceous layer. This layer also separates your cotyledon from the endospermic region. Okay, now in case of maize, you have only single cotyledon. Now it is referred to as scutellum. Okay, and now in your cotyledon, now you have a embryo attached. Okay, now just like dicot, embryo has two regions, plumule and radicle. Plumule gives rise to root system and your radicle gives rise to root system. Now if you look at the plumule, your plumule is covered with a outer covering, protective covering called coleoptile. Okay, coleoptile. How will you remember which layer is covering radicle and plumule? In coleoptile you have P here, and plumule also you have P. So coleoptile is the outer layer which covers the plumule. It forms a protective layer in the plumule. Okay, and radicle, which gives rise to root system, is protected by an outer covering called coleorhiza. R for radicle, and you have R in coleorhiza. So that's how you can remember. Okay, plumule is protected by a protective membrane called coleoptile and radicle by a coleorhiza. Okay, now your embryo is present in the tip region of this seed. Okay, so that's all about the structure of dicot and monocot seed. You should know how to draw the diagram and label its parts. Okay, what difference could you find between dicot and monocot seed? The difference was dicot seed had two cotyledons, and cotyledons were the one which provided nourishment to the growing embryo, right? But in case of monocot seed, you had only one cotyledon and nourishment was provided by your endosperm region. Okay, now in case of monocot seed, your cotyledon is also referred to as scutellum. Okay, so those were the difference between your dicot and monocot seed. So that's all about today's class. In next class we will be discussing in detail about germination.